Welcome back to Cityscape. In this episode of Secret People, we will cover Jennifer Doudna, an American biochemist known for the pioneering work known as Crispr. In a movie called Universal Soldier, Jean-Claude Van Damme plays the role of a super soldier who is the result of a secret army experiment. Unisols are faster, stronger, and feel virtually no pain. Aside from kick-ass action scenes, Universal Soldier spawned the imagination of its viewers. We all ask ourselves when such super soldiers will be a reality. Most of us guess the distant future, a period where technology will be unrecognizable. But our guests in this episode reveal super soldiers to be possible today. In fact, the first genetically engineered humans already exist and are alive and kicking. But we shall save this for later in the episode. As always, let's start with a brief background. Jennifer Ann Doudna was born on February 19, 1964, in Washington, D.C. At the age of 7, her family moved to Hawaii, where her father taught as a university professor, while her mother was a stay-at-home parent. When Doudna was in the 6th grade, her father handed her a copy of James Watson's book, The Double Helix. The plain language and explorative nature of this book inspired Jennifer to become a scientist. She went for a bachelor's in biochemistry at Pomona College, which she obtained in 1985. She then went to Harvard Medical School for her doctorate and earned her PhD in biological chemistry and molecular pharmacology. After her doctorate, Jennifer held several research fellowships, as well as teaching positions. She became a professor at Yale University, and later accepted a position at UC Berkeley. In 2006, Doudna was contacted by Gillian Banfield, a science professor at Berkeley. Banfield had been studying bacterial DNA and suspected the Crispr system to be a method microbes use to make their immune system more adaptive. Gillian reached out to Jennifer because she was one of the top experts on RNA. Jennifer would also meet a French biochemist named Emmanuelle Charpentier, who was studying how bacteria infect people. Together, these two gals would give the world the groundbreaking technology of gene editing. Now, let's take a look at the science. The adventure with CRISPR began by studying the adaptive immunity of bacteria. When a virus infects a bacteria, it injects its genetic information into the cell. The cell then replicates the viral DNA instead of its own. It turns out, however, that certain bacteria have found a way around this genetic hijack. Some bacteria have what's called a CRISPR system. This CRISPR system detects the viral DNA as foreign and stores this information in its locus. You can view the CRISPR locus as a sort of memory bank containing the DNA of all past viral infections. The cell then makes an RNA copy of this CRISPR sequence. This RNA molecule combines with a protein called Cas9. The Cas9 protein then searches the cell for a matching DNA sequence, and when it is found, the Cas9 protein cuts that sequence out of the double helix, in effect, removing the viral DNA from the cell. Here is a short clip of Jennifer explaining the process.
Now, just to give you a sort of another illustration of the way that bacteria are using CRISPR systems, I wanted to show you this video that illustrates how we imagine that CRISPR is operating in nature. So here's a bacterial cell that's being infected by a virus, injecting its DNA. And if the cell has a CRISPR sequence in the genome, it can acquire a piece of viral DNA into this part of its own DNA. And these are flanked by repetitive sequences, so it kind of signals to the cell that this is a special part of the genome. And when the cell makes an RNA copy of that sequence, those molecules, those CRISPR RNAs, can be chopped into units that each include a sequence that comes from a different virus. These RNAs turn out to combine with a second type of RNA called tracer to form a structure that can interact with a protein known as Cas9. Now that Cas9 protein is able to search the cell looking for molecules of DNA that have a sequence matching this CRISPR RNA sequence. And when that match occurs, the DNA opens up, the Cas9 protein cuts the double helix of DNA, and then these cut up pieces of DNA are degraded.
Pretty cool. So the CRISPR system is a way for a bacteria to remove viral DNA from its genetic sequence. To summarize again, the CRISPR locus serves as a memory bank containing the DNA of all past viral infections. This can then form a CRISPR RNA, which contains the DNA sequence of one particular virus. This CRISPR RNA also serves as a GPS finder for the Cas9 protein. When a Cas9 protein finds that particular sequence that matches the RNA, it cuts it out the double helix, and voila, the cell is virus-free.
I'm showing you here a cartoon of the Cas9 protein. So remember that that's being made in bacteria that have a CRISPR system. And it's literally a programmable protein. And what I mean by that is that it's got a program that is defined by this molecule of RNA called CRISPR RNA that has a 20-letter sequence derived from viruses, at least in bacteria, that can direct this protein to recognize a piece of DNA that comes from a virus because it has this matching sequence in the DNA. And importantly, this is a protein that requires a second type of RNA called tracer for assembly of this targeting complex. And when this thing searches the cell and finds a 20-letter sequence in DNA matching the CRISPR RNA, it's able to unwind the DNA double helix and allow Cas9 to make a cut in both strands of the double helix. You can think of DNA almost like a rope with two strands winding around each other. And this protein is like a cleaver that comes in and cuts the rope. And remarkably, this isn't happening randomly in the cell. It's happening only at a place that matches this guide RNA.
Now recall that Jennifer said Cas9 is a programmable protein. The question then is, can we program this Cas9 protein to cut whatever sequence we want instead of a viral one? The answer is yes, and this is exactly what these two gals have figured out how to do, giving us the powerful ability of gene editing.
Now this handle is the same in every RNA molecule, but we could trivially change this 20-letter sequence on the other end to direct Cas9 to a desired piece of DNA for cutting. And Martin did a great experiment where he literally designed 5 or 6 different single guide RNAs that allowed us to cleave a piece of DNA at places that we predetermined by simply inserting the desired sequence right here in the RNA. And when Martin did that experiment, that was for us the aha moment when we looked at each other and said, holy smokes, this is a programmable protein. We know how to control it, and we can make it introduce a double-stranded break in DNA at a desired place.
Now, you're probably asking, what happens after you cut the undesired sequence? Well, the cell has a natural mechanism to repair its DNA, but eliminating a sequence is not the only thing Doudna and her team can do. They can introduce a new sequence at the site where the cut was made. Not only can they introduce a new DNA sequence to the cell, they can make it inheritable, which would in fact create a new race.
I want to show you, this is a video that just helps you to imagine how this actually works. So here we are zooming into a eukaryotic cell, a plant or animal cell, where the DNA is inside the nucleus. And we're seeing Cas9 molecules with their RNA program searching through the DNA, looking for a matching sequence. And when this match occurs, this enzyme, this protein, is able to interact by unwinding the DNA. The RNA molecule is able to interact with one strand of the DNA inside the protein, and that triggers cleavage. So the DNA is cut, and then this protein Cas9 is able to hand off the broken ends of the DNA to repair enzymes in the cell that fix this break by processes that include making a small change to the DNA as shown here, but also sometimes to actually inserting a new piece of genetic information at the site of the break. So very quickly after we published this work in the summer of 2012, labs around the world began to adopt this tool for engineering and editing the DNA in cells and organisms of all types. And what was amazing was that it seemed to work robustly in many systems. So it wasn't unique to bacteria or even to human cells, but to everything else, plant cells, other kinds of single molecule, celled organisms, but also various other kinds of plants and animals.
Very powerful stuff, as you can imagine. This is easily one of the most groundbreaking discoveries in the history of biology. As Jennifer mentioned, this technique of gene editing works across all organisms: humans, animals, plants, bacteria, you name it. This means that CRISPR has applications across a large number of fields, from agriculture, medicine, and even warfare.
Tomatoes that can sit in the pantry, slowly ripening for months without rotting. Plants that can better weather climate change. Mosquitoes that are unable to transmit malaria. Ultra-muscular dogs that can make fearsome partners for police and soldiers. Cows that no longer grow horns. These organisms might sound far-fetched, but in fact, they already exist, thanks to gene editing. And they're only the beginning. As I write this, the world around us is being revolutionized by CRISPR, whether we're ready for it or not.
Now, early in the intro, I have mentioned genetically engineered humans already existing. They are in fact twin girls, Lulu and Nana. This experiment was done by a rogue scientist in China named Zhang Kui. He altered the embryo of a couple during fertility treatment, making the twin daughters incapable of being infected by HIV.
Chinese biophysicist He Jiankui, working largely in secret, had used CRISPR to knock out a gene called CCR5 in 2 early human embryos, then returned the embryos to their mother's womb. CCR5 plays a key role in HIV infection, and the girl's father was HIV positive. He said his motive was noble.
As a father of 2 girls, I can't think of a gift more beautiful and wholesome for the society than giving another couple a chance to start a loving family.
Zhang Kui's actions received widespread criticism, and the Chinese government suspended his research afterwards. Later, in December 2019, he was arrested by the Chinese authorities, fined 3 million renminbis, and sentenced to 3 years in prison. As for the twins, as of early 2022, authorities have declared both to be healthy. What do you think of this situation? Should Zhang Kui receive this harsh sentence? Or was he a brave misfit, willing to push the boundaries of science? Share your thoughts in the comment section.
Have no doubt, this technology will, someday, somewhere, be used to change the genome of our own species in ways that are heritable, forever altering the genetic composition of humankind. Just because we are not ready for scientific progress does not mean it won't happen.
Speaking of biology, one location you can check out is the Museum of Medicine in Paris. This medical archive dates back to the 18th century, showing the evolution of science and medical instruments. What's perhaps even cooler is that the museum is open to the public and is free to visit. Those of you who want to see the Museum of Medicine should save this location on a Cityscape app.
My wish is that this will provide a positive message to the young girls who would like to follow the path of science, and to show them that women in science can also have an impact through the research that they are performing.
Jennifer Doudna has received several awards for her groundbreaking discovery. At the top of the list, of course, is the Nobel Prize. Her and her colleague, Emmanuelle Charpentier, both received the Nobel Prize in Chemistry for, quote, the development of a method for gene editing, end quote. She also received the National Academy of Science Award, the Wolf Prize in Medicine, and many, many others. It is not far-fetched to say that Jennifer and her team have opened a new chapter in human evolution. How would you like to have blue eyes this year, and green eyes the next year? Or change your skin color like Michael Jackson did? Michael may have been far more ahead of his time than we realize. Some people will deny this technology, preferring to stay natural, as one puts it. This will very much create a situation seen in a favorite movie of mine called Gattaca. Which one will you be? A natural or an edited human? Speaking of Gattaca, the underlying theme of the movie is that people who do not meet the genetic or gender stereotype are just as capable as those who do. I find it ironic that this groundbreaking discovery, who will turn Gattaca into reality, was founded by people who do in fact defy norms. See you next time.